Hello, everyone. Good afternoon. Thank you for coming to this session about quality control in Metabolomics shared resource. My name is Xiaoyang Su. I'm running the Metabolomics shared resource at Rutgers University, New Jersey, and I'm honored to host this. Is it better now? Yeah, okay. Sorry about this. So again, welcome to this session about metabolomics quality control. My name is Xiaoyang Su. I'm from the Metabolomics shared resource at Rutgers University. So today I'm honored to be the chair of this session. And so before we get started, just want to reiterate a few housekeeping items. So please silence your phone, and the recording of this talk will be available through the ABRF website. I think everybody should have the mobile app by now. And just to remind you, after this conference, you will receive the evaluation survey, and that is very important for the future planning of ABRF. So please take some time to complete that. And so today, we're going to have three speakers, Ryan, me, Dr. Ying Liu, and me, and also Dr. Ryan Sheldon, who cannot be here, but send us a recording. And he will also be available after the presentation through Zoom for the discussion. Without further delay, please welcome Dr. Ying Liu for her presentation. So she's from the Georgia Tech Systems Mass Spectrometry Core. Please.
Thanks everyone for coming. And today, my talk is quality control. Can you hear me? Can you hear me? Okay. Thank you, everyone. And today, the topic of my talk is quality control in data acquisition and reporting in metabolomic core. So we all work in the core. We know how important is quality management in the core. When we're talking about quality management, usually we're talking about quality assurance and quality control. What is quality assurance? It refers to all the planned systematic activities implemented before samples are collected to provide confidence that a subsequent analytical process will fulfill predetermined requirements for the quality. Such activity will include formal design of experiment, certified and documented staff training, standard operating procedures for biobanking, sample handling, instrument operation, preventive instrument maintenance, and standardized computation workflow. What is quality control? Quality control refers to operational techniques and activities used to measure and report this quality requirement during and after data acquisition. Today, my focus is how we do the quality control. And this is typical workflow of on-target metabolomic study. And we start with sample collection. Then we homogenize samples and extract metabolites. Then we analyze the samples in the instrument, mainly LC mass spectrometry. After we get the data was processed by software, and we check the quality of features. After we have reliable features, we do annotation of the metabolites. And we have to have necessary quality control for each and every step of this workflow. When we do sample collections, we have to know the amount of samples. If it's cells, we count cell number. If it's tissues, we weigh the dry or wet weight of the tissue. If it's liquid form, we have to know the volume. Sometimes some lab may do protein concentration or DNA content, use that as amount to normalize the samples later. When we do sample homogenization and metabolite extraction, we have to have internal standard. We have to have process block and we have to make pooled samples used as quality control. For the instrumental analysis, we have to have solvent block, process block, quality control. We have to evaluate instrument condition. Sometimes we have to do system stability test. When we process the data, and we have to consider about how good is the peak shape, how the peak was aligned, what is signal-noise ratio, how about the variation of the QC and internal standard. When we do annotation of metabolites, we have to think about retention time match, accurate mass match, MS to the second, the spectrum match. And sometimes we wish we have the standard, we have did the study, and sometimes we have to consider about possible edits. Today, my talk will be focused on the quality control for instrument analysis, data process, and feature annotation. I'll talk about how we do the system suitability test for evaluation of mass spectrometry operations in the core. Before the data collections, we'll evaluate the mass operation by mass accuracy, mass resolution, sensitivity. We'll also evaluate LC operation by gradient profile reproducibility and internal standard retention time, internal standard peak shape, and width. After we did data collections, we'll evaluate the QC, and we'll also evaluate our samples. I'll give you one example to show you how we do the system suitability test in the core. So the LC system I use is Thermo Fisher Scientific Vanquish Horizon, UHPLC. The mass spectrometer is Q Executive HF. The column I use is C13 column. The sample is mouse plasma. And the analysis I did is untargeted lipidomic analysis in the positive mode. So the raw data was processed by Compound Discovery 3.3. First, evaluation of mass spectrometry operation by mass accuracy. So we do the direct infusion, use Pierce ESI positive ion calibration solution. And looking at the mass accuracy, the calibration solutions has 6 analytes. The 6 analytes cover from 100 to 2,000 mass range. So my untargeted lipidomic studies has the same full mass range. And you're looking at the figures, the M over Z deviations was estimated by the ppm change. So it is less than 3 ppm. The root mean squares is really small, so my instrument mass accuracy is really good. The second is mass resolution. Use the same positive ion calibration solutions by direct infusions into the instrument. And when you look at the figures, the maximum mass deviation is 1.0 ppm, so my mass resolution is really good. The third is sensitivity. The sensitivity of mass spectrometry was evaluated by measuring the signal-to-noise ratio from a single injection of the block contain my internal standard. So looking at the figures, top are 12 internal standard. They're heavy labeled lipids. Each one represents a specific class of lipids. And we have the baseline sensitivity. The baseline sensitivity was established by running a brand new column. Then this is my observed intensity for each of my internal standards, the observed noise for each internal standard. And intensity divided by noise, this is observed sensitivity for my internal standard. Compared with baseline sensitivity, the observed sensitivity has to be above 75%. I'm thinking I have a clean system. And you can tell the last line, everything's above 100%. So I do have a clean system, so my mass spectrometry will perform very well. the last one. Sorry. I want to go back. Yeah. Too much.
Somehow this slide
Too
is
sensitive,
eluding.
yeah.
We should use this.
Okay, try this. Yeah, this one. No. This one, yes.
Okay.
Yes. So next, I will do LC operation evaluation, first by gradient profile reproducibilities. So how we do the evaluation? We observe the pump back pressure change. If my column has any leakage or clog in my columns, it will cause the back pressure change. So I use a brand new column to establish the baseline of the back pressure, then compare with the baseline. And you can see the back pressure are relatively stable. The maximum pressure and minimum pressure are within the baseline. So my column will perform well. Second, I'll check the internal standards. I'll check the retention times and peak shape and width of my internal standard. So this is my 12 internal standard. You can tell they come at different times. I'll check the retention times. I'm looking at the peak shape. The peak shape is really sharp, very nice. And also check the intensity of each peak. They're really good. Then I convert this to a table. So I have the theoretical M over Z for each internal standard. I have the baseline retention time. I have the baseline FWHM. So these are done by brand new columns. Then I have observed M over Z. I have observed retention time and observed FWHM. So compare with the baseline, the mass errors has to be less than 3 ppm. And retention time difference has to be less than 0.2 minutes. And percentage of FWHM has to be less than 125%. And when you look at the tables, everything passed. So this tells me my mass spectrometry, my LC, my column, they all perform really well. Then I began to run my samples, my QCs, my black. After that, after I finish the run, first things I'm doing is the QC evaluation. For this run, I have 9 QCs. So I will overlay the QCs together. You look at the figures, the 9 QC overlaid really, really well. So I will observe like very, very minimum shift of retention times. And I also check overlay of intensity and the average intensity of my 9 QC. You can see the CV is really small, it's 1%. So the reproducibility of my QC from the beginning to the end of the run are really good. My instrument performance are excellent. Then next, I check the internal standards in my QC. The raw data was processed by Compound Discover. The Compound Discover will calculate the percentage of relative standard deviation for internal standard in all the QC. And you can tell from the table, and the 12 internal standard be detected in all the QC. And when you look in the last one circled by the green color, and the relative standard deviation is really small. So everything is lower than 50%. So my QC running is really good. Then I evaluate how is my samples. First, I'll check internal standard across the whole batch. I have 12 internal standard because of space, so I only show 2 internal standard here. The green dots is my black. The black, I didn't put any internal standard into it, so it's running at the beginning and end of the batch. And the orange dot is my QC, the blue dot is my samples. I put internal standard into my QC and into my samples. The QC was running at the beginning and end of the run. Every 10 samples, I have a QC. So when you're looking at internal standard PC, the intensity of the PC are very consistent in my QC and in my samples. And the black is true black. There's no internal standard of PC. When you're looking at the second internal standard DG, and the DG, intensity of DG is very consistent in my QC and in my samples. There's no DG in the black. Then I'll do the PCA models for all the detected features. This is PCA figure, and the red dots circled by a green circle, that's my quality control. And the green dots at the left side is my reference. The blue dots is my samples, the plasma samples. And you can tell the QC form very tight clusters in the middle. The reference form a relative tight cluster, and my samples are randomly divided into 2 clusters, divided by the PC1. And my QC, my reference, most of my samples are within the 95% of confidence line, and there's only 4 samples I treat as outlier. So by doing this, I didn't observe any issues for the data collection and data process of my samples. So the sample evaluation is really good. All the features I got are reliable. Then I go to the next step to do the feature annotation. Normally, when we talk about feature annotations, we'll see whether we have match of retention times, whether have match of accurate mass, and whether we have match of MS to the second spectrum match. And this is what we're doing previously. Currently, we added one more criteria. So when we run the samples, we either use QC, inject the QC with different injection volume, or if it's same cell type, we inject different cells number. So after we're doing this, for each features, we'll check intensity of features, whether the intensity of features proportional to the injection volume or to the cell number. I'll give you an example. This is HeLa cells extraction, and this was analyzed by Vanquish News coupled with thermal IDX. I inject different cell number, 150 cells, 750 cells, and 1500 cells. And I have 2 features. The first 3 features are assigned as PC. When you're looking at accurate mass, it's a very good match with the PC, and the mass error is 0.45. And you're looking at the left side, this checks intensity of this PC at different cell number. The intensity of the PC really proportional to the cell number. Then I believe this feature is truly from biological sample. And you look at the next one, and accurate mass match. The match error is only 1.82, but you're looking at the left, look at the intensity of these features. The intensity of these features are not proportionally to the cell number. So I do not believe these features coming from biological samples. It might be due to some combinations in my sample or in my system. So this is why we added new criteria. We have to check the feature, whether the feature's intensity proportional to the injection volume or to the cell number. So I'll show you how we do the feature annotation step by step. First, we have filtered feature. What do you mean filtered? So the feature intensity has to be 5 times higher than the background noise. The feature has to appear in half of my QC. The relative standard deviations of the features has to be less than 30%. So this I call the filtered feature. And within these filtered features, I'm looking for the features has name. For the feature has name, I'm checking whether the feature intensity was proportional to the cell number. Then this is my criteria 1. For the one match my criteria 1, I'm checking whether I see retention time match and accurate mass match. And this I call match with criteria 1 and criteria 2. Then with this, I check whether it's matched with criteria 3 or not. Criteria 3 is MS second spectrum match. So from here you can see I have high confidence features 629. These 629 features, some of them are lipids, some are small molecules. And within this 629 high confidence features, I have 670 high confident lipids features. And this lipid features was shown by class on the top figures. And these are annotated lipids in the positive mode and shown by different lipid class. This is how we do feature annotation. Basically, this is my talk. I'm talking about how we do system suitability test to determine the quality, the performance of instruments. Then we evaluate the QC, we evaluate the samples to prove the feature I got is really reliable. Then we put 3 different criteria to do feature annotations. So we have high confidence about the final features we have. So this is all about my talk. Yeah.
So that was fantastic work on lipidomics. I noticed that you can do as little as 150 HeLa cells, so that's very, very high sensitivity that you can achieve, and with so many lipids confidently annotated. So I think that's really fantastic, very impressive. Thank you. So next, I'm gonna switch to yes. So next, we're gonna switch to the recording from Dr. Ryan Sheldon. Before we get started on this, I really want to give him a special thank because originally we have a speaker, but because of a sudden visa issue, that speaker couldn't make it here. So that's where I asked Ryan if he can send us a recording. So Ryan is also the leader of the Metabolomics research group of ABRF, and we pretty much have monthly meeting discussing different issues in metabolomics core facilities. So if you're interested in this, please check the ABRF MRG website, and we welcome you to join us. So now let's get started.
Good afternoon, everyone, and greetings from Grand Rapids, Michigan. Hope you're all enjoying your time in Las Vegas. My name is Ryan Sheldon. I'm the director of the Mass Spectrometry Core here at the Van Andel Institute. The Institute is a biomedical nonprofit institute, and here at the Mass Spectrometry Core, we use mass spectrometry tools to answer biological questions using applications such as metabolomics, lipidomics, and proteomics. And so today I'm going to talk to you about some of the tools we have in place to keep track of how well our instruments are performing over time to make sure that methods are stable and that data is of the highest quality possible. So to start, I'd like to first acknowledge members of my team listed over here on the left. I'm really fortunate to lead a team of 10 really skilled technicians and scientists that keep the lab up and running and really make everything happen. I especially like to call out Abby Ellis, who's the lab manager, and this is her picture down here. She pulled these slides together today for me and really established a lot of the processes that you'll see presented here that we use to keep track of instrument performance. And so in the core, as I mentioned, we have a solid team of people. We also have 8 mass spectrometers, 6 of which are mostly dedicated to metabolomics and lipidomics, a couple over on the proteomics side. And you can appreciate from this list here that we do a lot of different things with our instruments. And it's not important to go into the details of what each one of these are, but they have very different needs as far as mass spec settings, chromatography, sensitivity, et cetera. And all told, the mass spec core processes over 8,000 customer samples every year, spanning over 150 projects. And so we have a pretty high volume core with the number of methods that we have here. And so you can really appreciate how important it is that we keep our instruments running and operational and at a peak performance as much of the time as possible. And so before I get into specifics on how we keep track of instrument performance, I want to highlight this principle that metabolomics and LC-MS metabolomics is really the beginning of a scientific journey. And so this is a project that I'm going to highlight that was started back in 2019 and 2020 by Kasia and Arem, who were postdocs in Rusty Jones's lab here at VAI. And what they asked was do T cells oxidize other substrates? So T cells are the soldiers of immune defense. They're part of your adaptive immune system, but they're generally studied in cell culture media that is primarily glucose and glutamine heavy for carbon source oxidation. So what they did is they actually went in and they traced these different carbon sources, including acetate, beta-hydroxybutyrate, lactate, and pyruvate. And to our surprise, we found that when we add these different substrates that we call PCS or physiological carbon sources to cell culture media, that the T cells actually prefer lactate, acetate, and beta-hydroxybutyrate for entry into the TCA cycle, which then spares glucose for things like nucleotide production and serine synthesis. And this paper was published in 2022, about 2.5 years after that initial data that I showed you on the previous slide, which really highlights the importance of having long-term stable methods so that we can iterate on projects and have data that are coming from the same platforms. But not only this, but this first project then spurred on several others that have been recently published over the last 1.5 years or so, and others are ongoing, looking at things like how beta-hydroxybutyrate will interact with histone acetylation, as well as how acetate and glutamine are also fuels for the TCA cycle, and what how this interfaces with epigenetics. And so now we're looking at data that has been collected on this overarching project for over the course of 5+ years. And so for this reason, it's really critical that we have long-term method stability and reproducibility for these ongoing projects so that we can continue to deliver similar results over a long period of time that can be added into a larger story. So how do we do this? First is we really invest hard into preventative maintenance. And this is something that a lot of labs, we tend to like to just ignore some of these things because we know that when we clean the instruments, things behave differently. And so it's, let's just run it until there's a problem and then we'll fix the problem. We've actually found in our experience that the opposite is true, and that the more that we can invest in preventative maintenance, the easier our lives are. And so these are some checklists that Abby has come up with, where we have on the left here a bi-monthly and every 3-month checklist of preventative maintenance items that we do, include cleaning filter lines and cleaning ion optics. We have a weekly checklist that we'll do where we're like cleaning the source, we're checking generator pressure, we're calibrating, making fresh solvents, running system suitability, which we'll talk about here in a moment. And then we also have a pre-run checklist where we make sure that everything is operating at optimal capacity, that chromatography is good, the mass spec is doing what it's supposed to do, and that we check all of these things before we run client samples. So I'm going to touch now on what we do with our system suitability standards, or what we call SSTs. So these SSTs are really just a set of samples or a set of standards that we use to verify that our system is working. And specifically, we use a mix of chemical standards that were hand-picked to be informative for us on our different methods. We have one that we use across the lab, so there's a mix of compounds that I'll show on the next slide. And these both allow us to look for problems with the chromatography system as well as with the mass spectrometer. And what this looks like in our lab is we have this list of compounds. And so we will make a stock of this at this final concentration, and we will aliquot these into single-use aliquots, and we make thousands of them at a time so that every time we're running a study, we can pull one of these aliquots out and have essentially the same reagent. And each one of these compounds is handpicked to be in this mix because it tells us something about one or more methods. For example, citrate is commonly a problem compound on one of our methods, and so we will specifically look at citrate to make sure it's behaving well both chromatographically and on the ionization side in the mass spec to make sure that our method is working properly before we go in to run the sample. So I'm going to walk through a few examples of how we use this data. So the first is we look for chromatographic stability. Often this is in terms of retention time and peak shape and peak area. So we generally look for if there's any shift in retention time over a half a minute. That's a bit of a guideline that we go by. It's not an absolute cutoff. Some methods and compounds we expect there to be more wobble from method to method, but generally a half a minute of shift is about the tolerance that we're willing to accept off of our optimal conditions. So for the case of this compound here, leucine and isoleucine, they're obviously isomers. And on this method, we're looking to get baseline or near baseline separation between these two and have roughly the same retention time as well as ion intensity. And you can see in these three examples that are spread out over the course of about 4 months that that's indeed the case. But then here is an example of when we get a right shifted or a later retention time. The peaks are a little bit shorter and fatter. And this really indicates to us that we probably have a chromatography problem, maybe a column problem, or the solvents weren't made quite correctly. And so that's a signal for us that we need to go do something. Similarly with ATP, adenosine triphosphate, this is another common problem compound that we have on one of our methods. And so we keep really good track of this. You can see generally we have a nice, beautiful peak shape at 16 minutes for ATP. But when the column starts to go bad, we know one of the first things that we notice is that we start to get a drop in sensitivity. Notice the scale bars on the left here on the y-axis that we're down about an order of magnitude here in these bottom two. And this is usually a harbinger for us that a column change is necessary before we continue. So it's really imperative that we get this information before we run customer samples. We also keep track of peak areas. So if peak shape and retention time look fine and we then have a drop in intensity, often that will point us to a mass spec issue. This is an example of glutamate, which we pay a lot of attention to, where we generally get this nice peak shape up into the E to the 7. And here we're down in kind of the mid E to the 6 range as far as ion intensity. And so this, again, might point us to a mass spectrometer problem. And then also we're not just looking at chromatography, we're also looking at the mass spectrum. And what we're looking for here is really just a qualitative assessment of how noisy this is. And we have images that show us what a good spectrum should look like. And then we compare that to what we're seeing. And so this is an example where we have kind of a normal full scan up on the top. But on the bottom, we have a lot more ions. It's very noisy around the baseline. And so this could point us to a problem either with our source being dirty or something else going on in the mass spectrometer. So going back through this list, what we have here is we had that retention time shift. And the two things that we'll check first in a case like this is, one, let's just remake solvents and see if it comes back. If that doesn't work, then we'll end up changing a guard column. If there's one installed, that just protects the column from other impurities in the sample. And then if that doesn't work, then we'll change the column. So we kind of go through the least expensive to most expensive option because columns are more expensive. So we like to make sure that solvents and guard columns aren't causing it before we change the column. But generally, one of these three things will fix this issue here. Similarly is true with the peak shape. You do start to learn your instruments over time. Like, for example, we know that when this happens to ATP, that really it's most likely not a solvent issue. The solvent issue may come with some like peak RT shifting, but when we get this drop in sensitivity, it does happen to be our chromatography. And I know that a minute ago I said drop in sensitivity is a mass spec thing, but we have noticed for some reason that we don't need to get into here that ATP specifically, when we get this dropping of the signal, that's actually a chromatography problem. And indeed, when we change to a new column, we were able to recover that ATP signal just fine. So I guess to summarize on that, there's some general conclusions that you can make from some of these metrics, like dropping in sensitivity, but you'll start to learn specific oddities about your instruments as you do these over time, and you'll be able to catch things faster and know what the solution is. When we start having losses in peak area and intensities that are not restricted to a single compound, so that's what's unique to that ATP issue, as I mentioned earlier, is that everything else in the method looks fine. It's ATP specifically is lost. When you have a mass spec sensitivity issue, maybe something is dirty, you tend to have a loss of all of the ions across the chromatogram. And so this is an example of glutamate, where there's a decrease in peak area. And then when in this case, we cleaned the source and cleaned the ESI needle, and we were able to recover that. Also pictured here as the front-end ion optic assembly of an IDX Orbitrap mass spec. Oftentimes, if the source cleaning doesn't go in and recover our sensitivities, we have to go a level deeper. This was also on our every 3-month preventative maintenance plan. So about 4 times a year at the very least, we're pulling this and cleaning it out. And sometimes if we're running particularly dirty samples, we'll have to do this even more often. And this is also the solution, source cleaning and ion optics cleaning for when we get a dirty spectrum. But we also included that I haven't mentioned is we'll also take the spray chamber and the source housing and make sure we clean that out really well. And so a combination of these things are able to both restore our sensitivity as well as get rid of some of the noisy baseline that we get from our mass spec and our system suitability runs. So when we run these system suitabilities, we need to check them. We need to check them again. But the most important thing is that if you're going to collect controls and system suitability standards, you actually have to look at them. And so this is something that we require our team to do, is that if you're going to run system suitability, we don't want to let the customer sample start running until we've actually evaluated those system suitability runs to make sure that the system is thumbs up. We're not just going to collect the data, let it go straight into the biological samples, and then come back tomorrow and look at the system suitability standards, because then if there's a problem, we still have to rerun the samples, right? Or maybe these are samples that we don't get a chance to rerun, right? So it's really imperative that you actually look at the system suitability data and evaluate it. And so if we're going to do that, we also have to have a rapid way to evaluate the system suitability to make sure it's working. And so the way that we do this in the lab is that we actually pull these data files when we collect them into Skyline, and we just have a Skyline file that we just continue to add data files to over time. So on, from left to right, we are looking at system suitability standards over the course of a year. And you can appreciate for this compound that we're generally pretty stable. Sometimes we have a drop, and we'll have a note associated with this data file that we probably did some cleaning, and then that recovered it to where we needed to be. We also keep track of retention time. You'll notice about right here, we had an increase in retention time. This is a time when we actually changed the column that we use on this method. And so we expected a little bit of a shift of retention time, but we have a note of that, and now we know what to keep track of going forward. You can see something similarly with PC 8080, which is in our lipidomics internal standard mix, system suitability mix. And you can appreciate maybe we have some more variability, maybe there's a different column here. And so we also make notes associated with these times when maybe things are looking higher or lower than normal. And you may be thinking, well, do you have a standard about when, like, is it within 20% or whatever? We typically use this more qualitatively. We do know that if we get drops down here, that these are usually something that we need to look at and try to correct. We also do some quality assurance checks using pooled samples. So these pooled samples are generated for every study. It's just a small aliquot of each sample in the study pooled together, and then it's ran every about 6 to 10 injections. And this allows us to see in the same sample, how is the instrument changing or responding over time? Because a lot of these studies can get quite large. This is an example of a lipidomic study in mouse skeletal muscle that ran over the course of nearly 50 hours. It was somewhere on the order of 100, 120 individual samples that were ran. And what you see here is an overlaid total ion chromatogram of pooled quality control samples that ran at the beginning, throughout the middle, and then at the end. And you can appreciate that the RTs and the signal intensities of the ticks are spot on, meaning that our system was incredibly stable over this 50-hour period. And so we have good confidence that the instrument was performing well. However, that's not always the case. This is another example of a metabolomics study that was a rather short one. It only ran for 9 hours. But you can see from the pooled quality control, black was first, red was second, green was third, and blue was last, that we had a real steady drop in intensity, even though our Ts looked fine. So this pointed, again, to a mass spectrometer issue because across the board we had a decrease in ion intensities. And the solution here actually ended up being that we had an issue deeper in the mass spec. Thermo came out and replaced or did a C-trap clean for us, which kind of gates the number of ions going into the mass spec. We rerun the samples, and sure enough, we have good run-to-run stability at that point. Finally, we also use internal standards. This is one example where we'll spike in isotopically labeled glutamate with 2,5-deuteriums on it. We'll spike this in at sample resuspension, and we'll keep track of that over the course of the run in every sample. And this is useful because, one, if you have a sample that just really is low or not, it's not present, that might tell you that you have a missed injection and it's worth re-injecting that sample. Or if you don't have a lot of signal intensity in the sample, but the internal standard's fine, it may tell you that you have a sample problem, but the mass spec is fine and that it injected fine. In this case that's plotted here, this tells us over the course of the run, it actually looks like we had a drop in signal intensity, and that when we did some source cleaning and rerun those samples, we were able to recover that, and the relative standard deviation was much improved. So, covered a lot of ground here, and this is something that we spend a lot of time thinking about. We've developed a lot of checks, and we've put a lot of things in place really based on lessons that we've learned the hard way. And so some conclusions from this that we can think about going forward is one, investing in routine maintenance really can prevent problems. The last thing that you want to do is just really hope that your instrument's going to hold on for this one last study. You run it, and then the data looks like junk, and then you're going to have to clean it and rerun it anyway, and then the samples are older, and maybe you don't have enough to re-inject everything, and yada, yada. So it's really just good to stay on top of your preventative maintenance, and this keeps your average downtime over the course of the year as low as possible. When you're designing system suitability and a strategy for which standards to use, it's really important that it's simple and it's informative and that you track it. It doesn't do any good to collect data that you're not going to use, so having overly complex standards really is not helpful, especially if you don't have an efficient way to look at it. So it needs to be simple, it needs to be informative, and then you need to track it so that you have a record of instrument performance and you know when things start to deviate. Pooled quality controls are really important. That gives you a bead on within study variance. Things that are going on with mass spec signal as well as chromatography with retention time and peak shape. You can evaluate those changes over the course of the run. Some people can use this for actual signal correction, using lowest regression or something like that. We tend to try to rerun samples if we can to get that more stabilized rather than using mass to normalize. It also allows us to see if the sample is degrading or changing in any ways. And then finally, internal standards are really useful. We like to spike them in at resuspension to check to make sure that the instrument injected, to distinguish between a sample versus an injection or a system issue. We also will add different internal standards at extraction, and that allows us to check for sample processing problems. And so internal standards can be really useful for a number of different reasons, depending on where you add them into the workflow. So with that, I'm happy to step over and take some questions. And if anyone wants to follow with me directly, here's my email address, and I would love to hear from you. So thank you for your time, and I hope you all enjoy the rest of your meeting.
It's really a fantastic talk from Ryan. So let me. Okay. Yes. Good point. Is it good? Yes. Okay. So number one, I think both Ying and Ryan covered a lot of very important parts of metabolomics quality control. So I think it may not be too helpful for me if I just repeat whatever they have said before. So I just want to share with you a few additional thoughts, a few things that we are rolling out in our facility to further improve the data quality and make sure we get reliable data. So of course, everyone start with a figure like this, just to give you the general the overview of the workflow. My point here is really the data quality control goes through every single step in this flow. But for today, I would like to focus on some specific issues. So number one, you already Ryan already showed you something like this. So especially for, I think this especially a problem for helic than to reverse phase, that over time you will have some retention time drift, and also you may have signal intensity drift. So our solution to this is mostly to use stable isotope labeled internal standards. So the reason we want to use internal standard, of course, the number one, as Ryan said, you want to know if this instrument issue or sample issue. So if we have a consistent amount of some compounds in every single sample, that number one can tell us, okay, the instrument is okay. And number two, we can use this to sort of correct the retention time drift if it's not super bad, so that you have to change a column. So when it comes to this stable isotope labeled internal standard, number one, you don't want this to interfere with your sample. So that's why we, like Ryan used deuterium labeled glutamine. This is not something that would present in your normal biological sample. So it's not an interference. And my idea is we want to have really a number of metabolites there to potentially cover the entire retention time range, because I want to use this to correct for some metabolite retention time drift. And also, we want to cover different types of metabolites so that we know, for example, it's not a single metabolite that is okay over hundreds of runs. It's really for the majority of the biological metabolites that we care about, they all have very good consistency across the runs. So these are some of my considerations in this. So this is the mixture we have come up with. So you can see a lot of these are amino acids, and that's because they're more available. But we do have a few other metabolites, like TCA cycle metabolites, because helic, we care a lot about central carbon metabolism. And also we have nucleosides that typically, and nucleobases that typically elute a little earlier in the helic runs. Again, our point is we want to have a group of compounds that are representative of actual biological samples and also span the entire retention time range. I want to talk to you about one thing that we learned the hard way. So when we put in the stable isotope labeled internal standards, initially we had a carbon and nitrogen labeled serine. We thought this was okay because we have the nitrogen on it. So even if we do a carbon labeling experiment, it shouldn't become an interference. Now, the carbon-labeled serine is a pretty important molecule in terms of carbon metabolism, central carbon metabolism. The reason is serine de novo synthesis is a metabolic pathway that is frequently amplified in a lot of cancer cells. So certainly people want to use glucose tracing to study how much serine is made de novo through the pH-GDH pathway. And when you do this, you give the cells or you give the animal, we can do this in vivo, you give the system carbon-labeled glucose, universal carbon-labeled glucose, and you're going to generate this carbon-labeled serine. Now, the problem with the first internal standard we came up with was that, number one, this was nitrogen labeled, so on mass spec level, you can totally resolve the de novo synthesized serine, which is carbon-13 labeled, and this internal standard. However, the problem is every tracer you buy from, say, Cambridge Isotope Laboratory or from Sigma Isotech, they have certain isotopic impurity. So here, the problem is the nitrogen-15 impurity is only 98%. Therefore, 2% of this tracer you put in is actually just carbon-13 3-labeled serine. So that is the metabolite that we do want to monitor from this glucose tracing experiment. So because of this isotopic purity issue, we have to switch to a carbon, deuterium, and nitrogen-15 labeled serine, which is luckily also commercially available. So this tells you that it really takes some experiments to find out what is the right labeled form that won't become an interference to your actual biological experiments. And another thing I'm very happy about our internal standard mixture is they're very affordable. So typically, if you buy internal standard mixture from Cambridge, I'm sure now they sell it, maybe even a few more metabolites here, but mostly amino acids. But the problem is very, very expensive. Now, we all know on mass spec, because it's so sensitive, you actually don't need a lot of analytes injected. And plus, the reason we want to use this is we want to have the internal standards signal level roughly matching the signal intensity we're detecting for the real metabolites. So we calculated the concentration for each of these metabolites, and based on how much we use, we calculate the cost of this, and we come up to the number of 3 cents per sample. So I think this relatively affordable, and if you're interested in this, you can take this recipe. So now I want to talk to the next issue, that is for highly confident annotation of our data. We want to have a very high quality in-house metabolite library. So we have spent a lot of time building this library in our lab. We actually have by now the 3rd generation of our in-house library. So you can see we gather about 400 metabolites, and we group them into about 20 different mixtures. And of course, you won't be able to have like isomers, leucine or isoleucine in the same mixture. Otherwise, you don't know which one comes first, which one comes later. So you have to design the standard mixtures very, very carefully to avoid isomers, and also if one compound potentially can come from the fragmentation, the in-source fragmentation of the other compound, you also don't want to put them in the same mixture. But after this, we can have this sort of plots. So for each metabolite, we have the chromatograms plotted under both positive and negative mode. Here, just to avoid the total overlap of peaks, I gave it a small offset on the x-axis. So the point is they have very consistent retention time, but still allow you to see that both of them have very consistent peak shape. And for each of the metabolite, we collect the information, of course, for the retention time and also for their signal intensity. So when we take a project, then if we have a handful of metabolites that are super important that we want to be able to measure, we can tell our users whether we should run a positive ionization mode or a negative ionization mode because we have the ionization efficiency in our in-house database. Another thing I want to talk about is, I think this is, we kind of see this as a frequent issue, but I think it does not get talked a lot. That is the chimeric MS spectrum. So what I'm giving you here is a real example. We're measuring this metabolite from a liver sample. And this top part is the actual MS2 spectrum we collected. Now, we know what this compound is, and we put the standard spectra here. So if you just look at this one, you would say this is a pretty bad match, okay? But at the same time, you also see, whatever you are anticipating from the standard spectra, the signal are kind of already there in the real spectra. It's just heavily contaminated by many other ions and many other interference. So what we can do is to use a sort of algorithm to clean the spectra so that we can generate, once we have this cleaned MS2 spectra, some people call this pseudo spectra, we also call this pseudo spectra, you can see this match perfectly with the standard. So this will give us a lot of confidence when it comes to metabolite annotation. So how do we do this? Now, normally when you collect MS2 data, if you use data-dependent acquisition, after you collect one spectrum, a lot of time you just put this into the dynamic exclusion and move to some other thing of lower abundance. So that gives you a better coverage of the metabolome. But also the point is, for everything, if you just get a single shot, there is no way you can tell if that fragment is from that metabolite or from interference. So what we're trying to do is, for each metabolite, we try to get multiple scans so that based on the parental ion and the fragment correlation, we can filter out the interference that don't correlate with the elution profile of the precursor ion. So here, this is example, this is the MS2 spectra, this is the parental ion in the MS1 domain. You can see for different fragments, if we plot their chromatogram for different chromatogram for different fragments, you can see some of them correlate perfectly with the parental ion. So these are the real fragments. Some of them don't correlate as well. They're just background ions, whatever. Even if you have the quadrupole doing the selection, somehow magically they're still there. So we're using this algorithm to filter out the interference fragments. Then we can have a pretty clean pseudo spectrum for this metabolite. Now, this is another real example, N-acetylglucosamine. So every spike you see here is from the raw MS2 spectrum. I'm coloring the real fragments in orange. So you can see that I certainly have some real fragments, but I also have some other very large fragments that don't poorly correlate with the parental ion, and they should be filtered out. So for things like this, I mean, this is good for sort of better metabolite annotation. But I think from our perspective, we want to give the users the full data. We want to tell them why we color a fragment either in orange or in gray. That's really because they have different correlation with their parental ion. So we came up with a plot like this. So here, each box, each rectangle you're seeing is a fragment. And on the x-axis is the intensity. On the y-axis is the correlation coefficient of this fragment with the parental ion. So we also rank them from the lowest correlation to the highest correlation. And you can see I really have a number of fragments that have pretty high correlation. Pretty high correlation with the parental ion. But things like 116 or 99, these are the two big peaks here. They have correlation coefficient below 0.2. So I think it's totally justified that we call this fake fragments and exclude them from the clean spectrum. So we are trying this also with DIA metabolomics. I don't know how many of you are doing DIA metabolomics. At least I know on thermal instrument, even though you can collect this type of data, but Compound Discover really doesn't handle this data nicely. But we can use our algorithm to process the DIA data, and we can cleanly identify the real fragments from this very complicated, very convoluted spectrum. So you can see here for cystathionine as example, we can see there are truly a few fragments that have very high correlation, while others have much, much lower correlation. So this is how you can identify the true fragments with the parental ion. So with that, I'm finishing my talk, and I just want to take this chance to thank my institution and also people in my facility, and also thank you. Okay, so now we're getting to the Q&A session. So I see Ryan is online. So Ryan, can you hear us? I probably. Hello?
Yep, I can hear you.
Okay, perfect. So yes, go ahead.
Xiaoyang, I love seeing your set of standards.
Can you speak to the mic?
Here, is it on? That should be better. Yes. Okay. I love your standard mix, and I wanted to know two things about including deuterium there. The first one is, do you ever have concerns about deuterium exchanging in your conditions? And I've never tried it in HILIC. Do you have any problem with retention time like you do with reversed phase, with the deuterium coming at a different place than the unlabeled?
Okay, so the question is, with deuterium internal standards, number one, do we have deuterium exchange? And number two, does it cause retention time drift? I don't think this has been a huge issue to us. We did monitor other labeled forms. My understanding is if we do detect a different, say if we put in a D6 internal standard and we're seeing D5 or even D4, that's really because of the isotopic impurity, not because of the deuterium exchange with the solvent. For the retention time drift, I never observed this on HILIC. Possibly this is a problem for reverse phase if you have heavily deuterated samples, but because it's supposed to be more hydrophobic than proton, right? But for HILIC, I have never seen this. Ryan? Yes.
Yeah. Can you hear me?
Yes.
I was just going to comment, hey Sue,
For some reason, that's Ryan. Hold on. This is a speaker issue.
Try now.
Hello, Ryan. Can you hear us?
Yep.
Okay.
I can.
Now we're putting you on speaker. Sorry, you wasn't on speaker. Okay.
Oh, that's okay. Hey, Sue, I was just going to comment that we have seen some hydrogen deuterium exchange. One example is with fumarate. If you buy D4 fumarate, you'll observe D2 fumarate because two of the hydrogens on either end are exchangeable. So it becomes compound dependent, and I would just run your neat standard before you spike it into anything to see how it behaves.
That's excellent. Yeah. Okay.
Thank you. This is Peng Gao from Northwestern. I'm running the Metabolomics course there. And first timer here, feel really great to find my people with core because the ASMS is just, too scientific. And right now, this thing, I have several questions after the 3 great talks. So first, I'd like to thank you for sharing that internal information about the internal standard, even have a great price list there. That's great. So I just want to know this talk going to be shared on the AB, on the conference website, right?
Correct.
Because I didn't have a chance to take a picture of that one. That's great. Second question, for all 3 presenters, yes, we talk about QC, QA, but customers, they send us samples, always worry about whether the sample from their end, whether the quality is good enough. I have tons of customers asking me, oh, I have done the extraction for lipids. I have done the extraction for metabolites. Is there a way for me to know whether the quality of the sample is good enough without burning through them? I have to pay, the customer have to pay the core for service fee because, that's the nature of the business. So I just wondering whether the 3 speakers, whether you have any information or doing something quality control, quality assurance before we even burn their sample. Thank you.
So number one, just quick information Ryan put in the chat box. That's why they do extraction in their own lab. So they can have more control over the sample quality. And I guess you also want to do this to prevent bad sample from contaminating your instrument, which may cause you a lot of trouble. So, but I guess it depends on how much manpower you have in your facility. But certainly, controlling the sample preparation, some metabolite extraction, I think will help with to solve your problem. Ying, do you have any suggestions?
Yeah, for us, we tried at the very beginning, some of the labs, they do the testing by themselves, and we do have a problem with our users do the extraction by themselves. So if they have to do it themselves by, decrease the cost, my suggestion is help them before they do experiment, tell them step by step. Yeah, and also give them standard and let them do it from the very beginning and standard with this back standard as well. But from our experience, let them to do extraction by themselves most times isn't very good.
All right. Thank you.
Hey, so my question was, we talked about how you guys keep track of all of your internal standards and recollect data. What is your data management procedures to then be able to take a look at this, longitudinal data of your standards? How do you handle the data internally?
I believe from Ryan's talk, he mentioned Skyline. I think Skyline has this capability, especially for monitoring the internal standard.
Oh,
yeah.
So do you just sync it to Panorama and keep track of it 24/7, or do you have anything else that goes more directly into your core?
Ryan said, yeah, just like what Ryan did, so we do make tons of standards at the same time, like allocate them, store them, and we will put, we also add the reference sometimes. So for the reference, they're the same material, so we do not worry about the matrix effect. So we'll periodically put them together and check it, to see what is longitudinal effect. But this do need a lot of work, that's for sure.
I might be a bit greedy and take more than one question, if you don't mind. A quick question to you. What is the algorithm that you are using for doing the cleaning of the MSMS spectra?
Sorry?
The algorithm, is your algorithm, is it published?
Yeah, well, we haven't published this.
Okay. So it doesn't have a name that we can go and find it somewhere?
If you're interested, we can maybe run a test project and just email me.
Okay. So maybe taking one more note, let's say. So this is for Ying and for Ryan, and thank you all for fantastic outlay of the QA process. But I didn't see any quality assurance on MSMS data, not in your talk, not in Sheldon's talk. Do you do anything about it to make sure that your mass spec is behaving in the MSMS cell? Because everything that I have seen was MS1 data before the analysis.
So the question is about, for example, system suitability test on the MS2 level.
Exactly.
For the MS2, we usually do the DDA, and DDA will take a long time to finish. That's why when we do the system stability test, we don't do it. But we do build a big library for MS to the second. So when we finish the run, when we check the features, we do check MS to the second. If something wrong, we know.
Yeah, I'm sure that you will know, but I'm talking about quality assurance before starting running, because I'm telling from personal experience. If the HCD cell is not behaving well, then you end up doing MSMS on all your data, and all your MSMS data is going to waste, and you'll need to go back, probably pool and redo the analysis, and with a helix column, goes into the same retention time or not. So that was my question, but apart from that, yeah.
So Ryan also put in the chat box answer that they do weekly calibration, which also handles the fragmentation tests. So I think you can get some information from there. And I think that's a good point that for the system suitability test, because we already have the standard, we could just collect MS2 layer of information and just make sure we have a good performance over there as well.
Okay, thank you very much. Three of you, thank you.
I'm going to do one more before I think we have to break. I was just curious on your performance assessments. How much is visual inspection versus having a specific defined criteria that you do computationally?
That's a good question. So for this quality control, how much of this is, how do we make the call, right? Ryan, do you want to take that question? You have the smiley face. So how do you make the call?
Yeah, so I guess the short answer is both. We find there's a lot of value in just having technicians look at ticks and EICs, and oftentimes that catches problems that we wouldn't have known before. We don't want to just look at a few variables. But then we also require that folks record for example, like the QC variance for the internal standard, and we want that to be less than 10%. We have some of those hard metrics that we look at to make sure things are going well. You can't necessarily use that for internal standards across all of the experimental standards or samples because you might run into a situation where you have different levels of ion suppression, so you kind of expect your variance to be higher in experimental samples, even if there's an internal standard involved. So you have to be a little bit more forgiving on those.
Yeah, I think, yes, that's right. Okay, so I think we're about to get done. So thank you again for attending this session. And if you have any other questions, you can find us. Thank you. Thank you, Ryan. Thank you for being with us.
Thanks, everybody.
What is